All right, we're going to take a little time and take a look at how we can compare the difference between mitosis and meiosis. If we were going to look at just some of the very basics behind them, we want to break down mitosis. Now, in order to do this, we want to be able to figure out what they're all about. Mitosis is taking a particular body cell. When we have a cell, and in humans, we have a total of 46 chromosomes. They're going to go through a cell cycle that is going to contain interphase. That's going to make this guy go up to 92 chromosomes, and then from there, break this down into two cells that are genetically identical with 46 in each one. Okay? In meiosis, we start the same way. We still have an immature sex cell, which is going to have 46 chromosomes. When it's going to go through the interphase process, we're going to get it all the way up to 92 again. But instead of just stopping here, where we're going to split it up into two that are 46, we're actually going to do this one more step. And each one of these guys will be broken into another cell. And that should have a total of 23 in each. These cells here are now considered haploid. These cells are considered having the full set diploid. And haploid versus diploid cells. So we're going to get a little bit into the basics behind each one of those here on. So what is the cell cycle? The cell cycle really contains a few parts. The first part is an I phase. I phase stands for interphase, that approximately 90% of the lifespan is spent in there, where we have three parts to it. We have G1, S, and G2. G1 stands for growth, or really gap, but first growth. Cell grows up, just like when you were a little kid, you grow up, so does a cell. Cell grows up. S stands for synthesis, which means making something. And in particular, we're going from DNA copies. DNA is going to get copied. And then finally, G2 is second growth, where we actually have now organelles copy. So organelles are going to be copied. Organelles are going to be things you learn in biology A, which are mitochondria, your ribosomes, endoplasmic reticulum, centrioles, all those other organelles are going to get copied. Now, one of the things we mentioned earlier is that when cells do get bigger, when they grow up, other products start to get limited. For example, simple diffusion. Things, as you get bigger, if you're thinking into a room, how quickly can you run from one side of a room to another? All right? In a smaller room, you can get there faster. In a bigger room, it takes you longer. So if as a cell break gets a little bit larger, cells get bigger and bigger and bigger, it's longer and longer for things to get into and out of a cell. So they get by with it. Instead of staying and continuing to grow, they decide to split. So in order to split, they have to go into an M phase. And M really refers to splitting the nucleus, and this M is mitosis. We're actually going to worry about body cells right now. Body cells is the same thing as somatic. Somatic cells. So in mitosis, there are three main steps. There are four main steps. There's prophase, metaphase, anaphase, and telophase. Prophase, we're going to have our particular cell. Okay? And in the cell, you're going to have a nucleus that was there is going to start to dissolve. Step one. Step two is that we had in interphase, we had a cell that in the nucleus, we had some jumbled up genetic material all up in there. Right? We went from our total of 46 chromosomes to 92 because DNA gets copied. And so I've got 92 chromosomes jumbled up in there. Now we're going to start to see those chromosomes condensing into little X's. They're not really in any order right now, but they're going to be condensing. We're in the metaphase. Now the nucleus is now dissolved, and those chromosomes now start to line themselves in the middle, and we have on either side a centriole connecting to the structure of the chromosome. Now, if I was to zoom in real quick on a structure here, look at the chromosome. This itself is considered a chromosome. In the S part of the interphase, we talked about DNA getting copied. We actually in the S part interphase get an identical copy, which is this one over here. And that copy is going to be held in the middle by this centromere. Okay, the centromere is going to hold them together. These two things right now, then, as they're being held together by the middle, are called sister chromatids. Chromatids. Okay. Now, if we can go back to our picture here, we're going to move our way from our structure. What we had, and just showing you, is that we had some fibers connecting to that centromere. Move our way into the next phase, anaphase. We have our cell. Okay. In our cell, we have this X that's now been pulled apart by either side of the pole. Okay. And then finally, we start to see telophase, where we get into 2. And in telophase, you're starting to see the genetic material that had been pulled to opposite sides are now in the middle. The nucleus starting to reappear. And then we are going to enter into our last phase at the very top, which is called cytokinesis. And what that job is to do is come down here and split those into 2. So we have 2 cells that are now completely identical, that have the same genetic material within it. Okay? So if we were to draw that out again, we had our one cell. We started out at a cell with 46 in there. After the S part of interphase, cells getting bigger. Now we have 92 chromosomes stuffed in there. And it's going to get through G2, still 92. We don't want to stay that big anymore. We need to split it into 2 cells. This looks like a 9. Okay. We are now going to split it into these cells. The splitting process here is known as mitosis, because we are splitting body cells. And what we want to have is 46 chromosomes here and 46 here. The same genetic. These two are identical. Identical, now called daughter cells. Okay. Next video is going to focus more on how the process of meiosis is going to take place and how that does differ in terms of our steps from mitosis.